Fine. Good morning, students. So this is final session of supplementary IPE revision because by today we can complete all the important VSAQs. So in the remaining chapters, whatever important VSAQs are there, today we are going to complete them. Okay. And hope that all these sessions you are using properly and getting benefited. So whatever the important questions being given in all these sessions, kindly learn them so that definitely you can get very good score. Keep a target and try to achieve for that. So at any cost this time you need to get very good result, and hope you will use all these sessions properly. Wherever you get doubt, you will go through the videos and use them in a right way so that you will be benefited. So let us go with the important VSAQs of remaining chapters. Already till yesterday we have completed all the important VSAQs. That is, I think so, till reproduction chapter, right? So till reproduction we have completed. Now let us see important VSAQs from reproductive health, genetics, evolution, and applied biology. In today's session, let us complete them. So now from reproductive health, I am telling you. So in the reproductive health chapter, the important VSAQ is what? Amniocentesis. So what is the question? What is amniocentesis? Okay. What is amniocentesis? And name the defects. Name the defects which can be identified through amniocentesis. Name the defects which can be identified through or by using this amniocentesis. So very simple. Actually, they have splitted the question and given. So with this, one can understand that by writing amniocentesis definition, you will get 1 mark. And by writing the defects that which can be known by performing this technique. So if you write the defects, you will get 1 mark. So totally 2 marks. So what is amniocentesis? So amniocentesis, it is a technique. It is a technique which is used to identify genetic defects. So genetic defects present in growing embryo or fetus. So present in the growing embryo or fetus. So that's all simple. So what is meant by amniocentesis? It is a technique which is used to identify the genetic defects. So genetic defects means what? So that means before the birth itself, whenever they are in embryonic condition or getting developed into fetus, so there are abnormalities with respect to the genes, or there are abnormalities with respect to the chromosomes. So it is because of mutations or so many factors are there again for these genetic abnormalities. So the genetic defects means that which are because of abnormalities seen in genes or the chromosomes, where they are differing with respect to the number of the chromosomes or with respect to structure of the chromosome. So if the number of the chromosomes are differing or the structure of the chromosome is varying, then they will only be responsible for causing what? Genetic defects. So amniocentesis is a technique which is used to identify the genetic defects, so that which are present in growing fetus or embryo is only called as what? Amniocentesis. Now, name the defects which can be identified by using this amniocentesis. And not only that, like previously, before government has banned this amniocentesis technique, even this amniocentesis been using for sex determination also. So that means in order to identify the type of sex, whether it is a male baby or female baby, that which is growing in the womb of the mother, generally amniocentesis technique is only being used. Okay. But however, now there is a statutory ban on this technique because of the increase in the number of female feticides. So that means killing of fetus in the womb of the mother. So that's why now this technique has been banned. But actually this technique is very helpful in order to identify the genetic defects or the chromosomal abnormalities. So what are the defects which can be identified now? So some of the defects based upon the chromosomal abnormalities like Down syndrome, then Turner syndrome, Edwards syndrome, Klinefelter syndrome. So like that, we can give list of diseases, but at least try to remember and write minimum 2 examples. Okay, minimum 2 examples you should write so that you will get 1 mark. So for this question, this is the answer which you need to present. So amniocentesis is a technique which is used to identify the genetic defects present in the growing embryo or the fetus, and the techniques, the defects which are identified are Down syndrome, Turner syndrome, and Edwards syndrome, and even Klinefelter syndrome also you can write. So that is about amniocentesis. So then next, after this, coming to another important question. So from reproductive health, that is, what are the measures one has to take to prevent contracting STDs? So what are the measures one has to take? Next question is, what are the measures one has to take? One has to take to prevent contracting STD. What is meant by STD first of all? So STDs are sexually transmitted diseases. So STDs are what? Sexually transmitted diseases. So they are asking, what are the measures which you take in order to prevent or avoid getting STDs? That is the meaning of that question. So tell me how STDs can be prevented, sexually transmitted diseases can be prevented. Actually, STDs is because of what? Sexual contacts with the unknown partners or illegal partners, okay, or with multiple partners. For example, if they have any sexually transmitted disease, because of having sex with them, what happens? Even the other partner also easily will be getting the sexually transmitted disease. So that's why what should be done? Avoid sex with multiple partners or unknown or illegal partners. Okay. So by avoiding sex with multiple partners or illegal partners, obviously sexually transmitted diseases can be prevented. And not only that, so whenever safe sex is not being done, so then also there is more chances for getting STDs. So that's why, so use condoms to have safer sexual practice. Okay. And STDs, no, however, immediately they can't be recognized, because for most of the STDs, the symptoms would not occur immediately. So that's why what happens, definitely the person would not be knowing that he has been affected with sexually transmitted disease. Okay. But whenever you get a doubt, or whenever, no, with mild symptoms only, no, when you are getting mild symptoms, then immediately go to doctor and get confirmed with the disease. And if you start taking treatment, then definitely sexually transmitted diseases could be avoided initial stages only. Understood? So that is identifying the symptoms and getting treated at initial stages of STDs. Okay. See, generally, no, even though the people, they have doubt of STDs also, but they will have this, social stigmata. What is meant by social stigmata? Like feeling, shy or ashamed of going and saying that I have so-and-so symptoms in so-and-so genital areas, like that. So that is very wrong, because once the STDs, no, they become final stages, then what happens? So they will be very tough to cure. So that's why always STDs need to be identified at the initial stages and need to be treated. Okay. So like this, at least 3 points you are supposed to write. Like, what are the measures one has to prevent contracting STDs? So that is avoiding sex with multiple partners or unknown partners, and using condoms in order to have safer sexual practices, and identifying the symptoms of STDs and getting treated immediately at initial stages. So these are the measures one has to take in order to prevent or contracting STDs, that is sexually transmitted diseases. Okay. The next, coming to other questions. So other questions. Then let us go with the genetics chapter. Let us see the important questions of genetics chapter. What are multiple alleles? And the next question, what are multiple alleles from genetics? Multiple alleles. Let us see important questions from genetics. What are multiple alleles? So now, important VSAQs from genetics. What is the question? What are multiple alleles? What are multiple alleles? So what are multiple alleles? What is meant by allele, first of all? So alleles are the alternative forms of a gene. Alleles are what? Alternative forms of a gene. For example, imagine that, if a plant character, when you take, like the character like height, you know that definitely there will be specific gene for each and every character. And now the alternative forms or the alternate traits of a character is only called as what? Allele. So that means what are the alternate characters for the height? So the alternate characters are, one allele may be coding for tallness and the other allele may be coding for dwarfness. One allele may be coding for tallness and the other allele may be coding for dwarfness. So that means the alternative forms of a trait are only called as alleles. So generally for every gene, mostly there will be two alleles, the alternate forms of a gene. But generally what happens, with respect to some cases like multiple alleles, for every gene now, there are many alleles. Okay, if you consider a gene coding for a character and if it is having many number of alleles, okay, or multiple alleles, then that is only called as what? Multiple alleles. The best example is, let us take ABO blood grouping system. So if you take ABO blood grouping system, actually here the gene is one. Gene is one. The blood group coding gene is one. But the alleles are multiple for that character. Or the alleles are multiple for the gene. For example, so that means the concerned person may have A blood group depending upon on the RBC membrane if the A antigen is present. On the RBC membrane if the A antigen is present. So that means the allele that which is responsible will become what? So that is A blood group coding allele. Okay. So like this, multiple alleles are those that which have many alternate forms for a gene. So many alternate forms for a gene are only called as multiple alleles. And the best example for that is what? ABO blood grouping system. So where it may be having IA or IB, okay, or otherwise it would be IAB. So that means the genes that which are responsible for coding. For example, if a person is having A blood group, so then he would be having allele like IA, or it may be IA-IO. Or if a person is having B blood group, then he may be having IB, or it may be IBO. Or if the person is having AB blood group, so then he may be having what? IA, and another one is also what? IA or IB. So like this, depending upon the type of the allele they are containing, so the person's blood group can be identified. So like this, ABO blood grouping system is best example for multiple alleles. And even skin color. So skin color is also another example for multiple alleles. So skin color is also one more example for multiple alleles. So for multiple alleles, what you are supposed to write? When there are many alleles for a specific gene coding for a character, then they are only called as multiple alleles. And example is what? ABO blood grouping system and then pigmentation of skin. So that is about multiple alleles. So then next coming to I think so because coming now. So what is the reason for the anemia and the jaundice? Because the antibodies has passed through the placenta and started to destroy the fetal RBC, so that no, the concerned baby, no, the newborn baby will be having anemia and then jaundice. And not only that, as more and more RBC are getting destroyed, so that what happens? The fetus in the fetal liver. So the fetal liver will start to produce more and more RBC. So which is only called as what? Erythroblastosis. What is the meaning of erythroblastosis? Increase in the number of RBC. Okay. So increase in the number of the RBC. Abnormal increase, not simple increase. Abnormal increase in the number of the RBC is only called as erythroblastosis. So that almost abnormal RBC are formed now in the fetus. And the babies who are developed due to this condition, so they are only called as what? Blue babies. So the babies formed due to erythroblastosis fetalis are called as blue babies. Clear, understood? So Once again, I'm repeating. Erythroblastosis fetalis is a condition where RH positive man marries to a RH negative woman. And in the first pregnancy case, if the baby is RH positive, then mother will become sensitized. Because during the delivery, the umbilical cord is cut. Some of the fetal blood would be mixed with the mother's blood, so that mother will become sensitized and produce antibodies and memory cells. So you know that memory cells are retained in the blood. And again, during second pregnancy, if the fetus is again RH positive, now these memory cells will differentiate and produce antibodies, so that antibodies will cross the placenta and start to destroy fetal RBC, so that newborn babies would be having jaundice and anemia. And not only that, as more and more RBC are getting destroyed, the fetal liver will start to produce more and more abnormal or immature RBC. So which is only called as what? Erythroblastosis fetalis. Clear? So like this, these are the two important questions which you may get from genetics. So one is, what are multiple alleles? And the second one is, so the next coming to evolution chapter. Let us see some of the important questions from evolution. So, atavism. Define atavism with an example. Atavism with an example. Now, important VSAQs from evolution. Organic evolution chapter. So, define atavism with an example. So, this is also simple and easy question. You should remember, you already would be knowing about appendix, which is a vestigial organ, right? So, like that, in the human body, there are many vestigial organs. Okay, there are many vestigial organs. So, what happens? Atavism is a condition where some of the vestigial organs again would start appearing, reappearing, or functioning in a developed condition. For example, you like a human baby is born with a tail. You know that humans, they do not have tail, right? So tails were there previously in the ancestors. But slowly what has happened? So as the usage of the tail has been reduced depending upon the environment or the conditions where they are living, so slowly the tail has been started to disappear. So but now the baby born with the tail is only called as what? Atavism. So atavism is reoccurrence or reappearance of some vestigial organs in the body, in the human body in a developed condition. So in a developed condition, so this is only called as what? Atavism. So the best example for this is what? Human baby born with a tail. So human baby born with a tail. Okay. So this is your textbook example. So definitely you should write this only. So reoccurrence or reappearance of some of the vestigial organs in the human body in a developed condition is only called as what? Atavism, and with example, so that you will get 2 marks. Then coming to so next question, define biogenetic law and giving an example. So what is meant by biogenetic law? So this biogenetic law can also be called as theory of recapitulation. So define biogenetic law and give an example. So what is meant by biogenetic law? Biogenetic law can also be called as theory of recapitulation. So that which is proposed by Ernst Haeckel. So who proposed this theory of recapitulation? Ernst Haeckel has proposed the theory of recapitulation, where according to Ernst Haeckel, so ontogeny repeats phylogeny. Ontogeny repeats phylogeny. So this is very important statement in the evolution. Yes or not? What is meant by ontogeny? Ontogeny means the developmental history of an organism. It includes or repeats the evolutionary history of an organism. So the developmental history of an organism, it repeats what? It repeats the evolutionary history of an organism. So this is only called as what? Ontogeny repeats phylogeny. So ontogeny is the developmental history of an organism. Developmental history of an organism which includes the evolutionary history of organism. What is the meaning of this? Which includes the evolutionary history of organism. So this statement you can explain best with an example. For example, let us take frog. So the tadpole larva of frog, it consists of what? Gills. Right? For the exchange of the gases, for the process of the breathing, the tadpole larvae of frog consists of gills. And what happens during metamorphosis? So that means during metamorphosis, where there will be drastic changes for the larval form to get converted into adult. So the gills will disappear and the lungs develop. Yes or not? So, but actually gills is which character? Presence of gills is ancestral character, that which is seen in fishes. Yes or not? So presence of gills is the ancestral character. That means ancestral character means evolutionary character. So evolutionary character has become part of the life history of an organism. So that's why it is called as what? Ontogeny repeats phylogeny. Example, tadpole larva of frog containing gills. So which is ancestral character, that which is seen in what? Fishes. Clear? So that is the meaning, ontogeny repeats phylogeny. So very, very, very important question. Many times they have given this question. Define biogenetic law with an example. So definitely must to learn. It is also called as theory of recapitulation, proposed by Ernst Haeckel. And biogenetic law states that ontogeny repeats phylogeny. The meaning of that is developmental history of an organism includes the evolutionary history of an organism. And for that you are supposed to write an example like tadpole larvae of frog containing gills, which is the ancestral character seen in fishes. So that is only called as biogenetic law. The next coming to other important questions. Define founder effect and give one example. So this is also one of the important question. Define founder effect and give one example. So next question from evolution chapter only, important VSAQ. So define founder effect and give an example. Define founder effect and give an example. So what is meant by founder effect? So whenever a small group of individuals are getting isolated, so because of some accident or some natural calamity, whatever has occurred, only if a small group of individuals are left over, and if they begin to form a new population in a particular area, now what will happen? They will become the founders. They will become the founders. And next, whatever the individuals come, they will be phenotypically and genotypically similar to these individuals. And such kind of effect is only called as what? Founder effect. So this, in a simple way, you can understand that. Let us imagine a population like blue colored beetles are growing. Let us imagine. Okay. In a particular grassland, blue colored beetles are growing. So what happened? Because of some variation, okay, because of some variation, what happened? The green colored beetles also started to develop due to mutation, and because of the appearance of the variations, the blue colored beetles, imagine that first initially blue colored beetles were only there in the grassland, and due to some variation, what happened? Even green colored beetles also started to appear. Okay. Now what happened? Slowly blue colored population was increasing and green colored population was increasing. Okay. So, but actually green colored population has survived more. And whereas blue colored beetles were easily identified by birds, they were easily identified by birds and started to get eaten away. So that's why blue colored population were slowly started to reduce. Okay, like this the condition was going on, because now you know that green color beetles cannot be identified easily on the grasslands. Okay, so or something like if there are plants which are green in color with green colored leaves and all. And imagine that one day a herd of elephants has come. Okay, a herd of elephants has come, and these elephants, imagine that they had a ferocious fight between them, where in that ferocious fight, okay, by chance, what has happened? As blue colored beetles are already less in population because of eating away by birds, now in that accidents, all the blue colored beetles got destroyed by stamping by these elephants, which are very angry or ferocious. And imagine that only green colored beetles, some of the green colored beetles got survived. Now what happened? Now these green colored beetles only become what? Founders. They will only become the founders, and the next species whatever come, or the next individuals whatever come from these beetles, so now they will be both phenotypically and genotypically, phenotypically and genotypically, they will be similar to the founders. They will be similar to founders. Yes or not? Because the next population or the young ones, whatever come, they will be similar to that of their parents, both phenotypically and genotypically. Such a kind of effect is only called as what? Founder effect. Clear? Understood? So imagine that only blue colored beetles were there, and due to variation, slowly green colored beetles also got to develop. But green colored beetles were not identified by the birds easily, as they are growing on the leaves of the plants which are green in color. But blue color were identified easily. So that's why blue colored beetles were slowly started to eaten away by the birds, and their population was decreasing. And one day what has happened? A herd of elephants has come, and they had a fight between them, where in that accident all the blue colored beetles got destroyed and only some green colored beetles got survived. Now they will become the founders, and whatever the individuals which will develop or raise from these, now they will be phenotypically and genotypically similar to this. So that's why it is called as founder effect. Okay, so founder effect is small group of individuals getting isolated and start to form a new population, which are called as founders. And the individuals next, whatever come, they will be similar to that of the parents, which is only called as what? Founder effect. So that effect is only called as founder effect. So like this, from evolution chapter, 3 questions definitely everybody should learn. One is biogenetic law, and the other one is what? Founder effect. Okay. And the previous one is atavism. So these 3 questions definitely you should learn. Then last, coming to applied biology unit, the last unit. Let us see some of the important questions from this chapter. In this chapter, some 4 questions are there which are very important. Define the terms layer and broiler. Define the terms layer and broiler. So what are layers and what are broilers? Very simple, no? Layers are the birds that which are meant for the production of eggs. Okay. Whenever you are raising the birds for the purpose of eggs and meat, and specifically the birds which are reared for the production of eggs, such kind of birds are only called as what? Layers. And if the birds are reared for the production of the meat, such kind of birds are called as what? Broilers. So layers, the birds reared, or the birds which are reared, and if they are meant for the production of eggs, if the birds are raised for the production of eggs, then such kind of birds are called as layers. And what are broilers? Broilers are the birds reared, so for the production of meat. So mainly they are reared for the production of meat. So for each point, one mark. For layers, when they see eggs, immediately they'll put 1 mark. When they see meat, immediately they'll put 1 mark. So that easily you can get 2 marks. Okay. The next important question, define apiculture. Define apiculture. So what is meant by apiculture? Apiculture is rearing of honeybees and the maintenance of the beehives. So whenever honeybees are reared commercially as a profession, because you know that so many products we get from honeybees, like honey we get, bee wax we get, propolis, and bee venom is used in the treatment of arthritis. Already I told you previously also. And even bees are a very good pollinators, so that the crop yield can be improved. Yes or not? So like this, many uses are there from honeybees. So that's why if any farmer will take the profession of rearing these honeybees, so that rearing is only called as what? Apiculture. So apiculture is rearing of honeybees, and maintenance of beehives for the production of commercial products from honeybees. For the production of commercial products from honeybees, like honey, wax, etc. So you can write whichever you know. So like honey and bee wax, etc. So this is only called as what? Apiculture. So apiculture is rearing of honeybees and maintenance of beehives. Okay. The next question from this applied biology chapter. Mention any 4 fish byproducts. So this is also a very important question. Many times they have given in the previous exam papers. Mention any 4 fish byproducts. Okay, mention any 4 fish byproducts. In this, you know that shark liver oil and cod liver oil, they are the good sources of vitamin A and D. So, shark liver oil and cod liver oil. So, they are the good sources of vitamin A and D. And the next product which we get from shark is shagreen. What is meant by shagreen? The dried skin of shark. The dried skin of shark is only called as shagreen, which is again used to prepare many products. Okay, that means the skin of it, it can be used something like a leather to make, some kind of quality products like bags, okay, or a certain kind of shoes, that which can be called as what? Shagreen. So shagreen is a dried skin of the shark. And apart from that, fish guano is a fertilizer. So fish guano is a fertilizer. So the fish guano can be used as a fertilizer. And one more important product which can be prepared is Ilein glass. So Ilein glass is also one of the important byproduct prepared from what? Dried air bladder of fish. Dried air bladder. So what is the importance of air bladder in fishes? They help them in providing buoyancy, okay, floating. So they are prepared from dried air bladder of fish, that which is used in the clarification of wine. So this Ilein glass is used in the clarification of wine. So like this, 4 important byproducts of fishes you can write. First one is what? Shark liver oil, shagreen, fish guano, and then Ilein glass. Shark liver oil and cod liver oil are the good sources of vitamin A and D. And shagreen is a dried skin of shark, which is used to prepare many products like bags. And fish guano, it is used as a fertilizer. And Ilein glass is actually extracted from the dried air bladder of the fish, which is used in the processing of wine. That is clarification of wine. So these are the 4 important fish byproducts. The next one, hypophysaceation. This question also, so many times they have given. Because you know that applied biology chapter, mostly 2 mark questions, like either 1 or 2 will be given every time. From applied biology chapter, if they are giving 1 SAQ, then you can expect 1 VSAQ. If they are not giving SAQ, then 2 VSAQs you will get from this chapter only. Okay. So last one, explain the term hypophysaceation. So what is this hypophysaceation? So during artificial breeding method of fishes, breeding method of fishes, what is done? Pituitary gland is collected. So you would be knowing pituitary gland, which is the endocrine gland present in the brain, right? So this pituitary gland is collected and crushed. Pituitary gland is collected and crushed. And from this pituitary gland, you know that actually pituitary gland secretes hormones like gonadotropins. So what are gonadotropins? Gonadotropins are FSH and LH. FSH is follicle stimulating hormone and LH is luteinizing hormone. So these gonadotropins are extracted. Okay. So these gonadotropins are extracted and they are introduced into brood fishes. So they are introduced into brood fishes in order to produce healthy seed production. So that means in order to get many seeds of fishes. So what is seed of the fish? The seed of the fish is something like a parent, the precursor, raw material, in order to produce high quality fishes again. So like this, the gonadotropins are extracted and introduced into the brood fishes so that whatever the eggs are laid, they will be collected. that which can be called as what? Healthy seeds. And for the future purpose, they will be using them. So that is only called as what? Hypophysis. So hypophysis is during artificial breeding of fishes, the pituitary gland is crushed and gonadotropins like FSH and LH are extracted and they are introduced into the brood fishes where the eggs will be collected again and used for the future purpose, called as hypophysis. Okay. So these are some of the important questions. And the last question, what is popularly called guardian angel of cell genome? Okay. What is popularly called guardian angel of cell genome? So this is also very important question. Last question of applied biology. What is popularly called? What is popularly called guardian angel of cell genome? Guardian angel of cell genome. So the P53 gene is called as guardian angel of cell genome. So the first point which you should write is what? P53 gene. So the P53 gene is called as guardian angel of cell genome because it helps in maintaining the integrity of DNA. Because it helps in maintaining the integrity of DNA means what? Protecting the structure and dynamic functioning of the DNA, which is called as integrity of DNA. And generally this P53 gene, it acts as a checkpoint. It acts as a checkpoint. So thereby controlling cell division or cell cycle. So that it prevents what? So that it prevents the cancer or tumor. Okay. So like this P53 gene, it acts as a checkpoint in the process of cell cycle or cell division, particularly at the G1 phase. At the G1 phase, it acts as a checkpoint. So thereby it helps in repairing the damaged DNA. Thereby it helps in repairing the damaged DNA. So that means it is avoiding the mutated things, thereby not allowing to develop the cancer. So P53 gene is called as guardian angel of cell genome. It helps in maintaining the integrity of DNA, where it acts as a G1 checkpoint, thereby not allowing the cell cycle to proceed. Before allowing the cell cycle to proceed, it is repairing the damaged DNA. Okay, so thereby avoiding the development of the tumors. So that's why it is called as guardian angel of cell genome. That means protecting, taking care of, guardian. So that's all. All these are important VSAQs. Totally 40 to 45 VSAQs I have told you. Daily if you study 5 also easily you can complete. Okay. And remember only 4 LAQs. Male reproductive system with diagram, female reproductive system with diagram, then structure of heart and working of heart. So 4 LAQs if you learn, definitely 2 will come. And SAQs, almost some important 15 SAQs I have given. So go through all of them. And VSAQs, nearly 40 VSAQs we have discussed. At least in this, if you learn some 20 or 30 also, you will be able to write minimum 3 to 4. Minimum 3 to 4 definitely you will write, so that you can get a good score. So that's all. Thank you for giving this opportunity. Kindly go through the videos, use the sessions properly, learn all the questions, because we made it in a simple way, even the complicated answers also. So use in a right way. If you have any doubts, you can ask. Otherwise, we will end up the session.